In the name of God, our subject for this session is about the cell. You know, the cell is the smallest structural and functional unit in the human body and other creature. In this session, students should know about the microscopic structure of the cell membrane, function of the cell membrane, and cell signaling in the cell membrane. You know, in the world, there are two types of cells, prokaryotic and eukaryotic cells. You know difference between of them. Prokaryotic cells, no nucleus, no membrane-bounded organelles. The organelles are spared in the cytoplasm, and they have 1 to 10 micrometer size that include the bacteria. That the cell have cell membrane and also the cell wall. But eukaryotic cells, that EU means the good, have a good nucleus, very sharp and distinct nucleus that restricted by a membrane. That they include many organelles, and the size of them is very variable. And all other cells in all of the creature include of these cells. But all of the cell in the human body derived from zygote, that zygote after some mitosis division formed a aggregation of the cells that named morula, that each of these cells can differentiate to one embryo and one person. In the development and differentiation, the cells divided in two groups, inner cell mass and outer cell mass. That inner cell mass finally differentiate to the body of the human. These cells in the in vivo condition in the embryo finally differentiate and form different cells with different structure and function. But in the in vivo, these cells that named embryonic stem cells, with adding of some inducers, can differentiate to other cells with different structure and function. As you see in this slide number 6, we can define and describe the cell differentiation as a procedure that the one cell as a stem cell change its function and structure and finally formed some cells with different structure and function in the body. Cell differentiation occurred in most of the cells. That in this process, the shape of the cells and the function of them change, and each of them, for example, neuron and sensory cells, have some process in their structures, and they can conduct some action potential. And you can see in this table different cells with specialized structures and also specialized activity and function. After this introduction from slide 18.8, we want to know about the cell, the structure of the cell. The cell, all of the cell that differentiate in cell differentiation and have different structure and function, but all of them have three main parts, three main components. Cell membrane, that is a cell membrane, that is a membrane that restricts the cell. Cytoplasm, that include the cytosol and also organelles, and the nucleus that mostly located in the center of the cell and control the all function of the cell. That we want to know one by one in this session. Cell membrane, also named plasma membrane or plasma lemma, include two layers of phospholipid. That when we do tetraoxide osmium staining, we can see as this structure here that we can under electron microscope, we can see the phospholipid bilayer as two dark and white layer that on tetraoxide osmium staining named unit membrane, the cell membrane. Cell membrane visible with electron microscope. But if we do the special staining such as periodic acid shift, maybe we'll be able to see the cell membrane by light microscope. The thickness of the cell membrane in different cells is between the 7.5 until 10 nanometer. Cell membrane include phospholipid bilayer, proteins, cholesterol, and oligosaccharide chain that we know about them in detail in other slides. Phospholipid bilayer, as you see here, it includes two layers of phospholipid that each full phospholipid molecule contain a hydrophilic head that is toward the cytoplasmic space or extracellular space. And in the middle part or center part of this layer include the two hydrophobic chain that they are located in the middle and are adjust together. The type of phospholipid and the location of them is very, very important in physiological condition. For example, in the red blood cells, because the easy access to these cells, the cell membrane of these cells is well known. The two types of phospholipid, sphingomyelin and phosphatidylcholine, is more in outer half, and in the inner half, the phosphatidylserine and phosphatidylethanolamine is more in the inner half. These type of phospholipid in each cell are special, and the amount and location of them is very important. If the amount of them change or the location of them change, they can result in some disease. The other component that we can see in the cell membrane are proteins. There are two types of proteins in the cell membrane: peripheral proteins and integral or transmembrane protein. As you see here in this schematic picture, the transmembrane or integral proteins interact with lipid bilayer, interact with both of their layer, and mostly they act as a carrier or transporter in cell membrane. But the peripheral proteins are located just in one layer of the phospholipid, in the outer layer or inner layer, that give to asymmetry to the cell membrane. The peripheral proteins in the outer layer, some of them, not all of them, some of them join to sugar chain and form the glycoprotein, and the peripheral proteins that are located in the cytoplasmic side of the cell join to the cytoskeleton and involved in the maintenance of the shape. Oligosaccharide chain attached to the phospholipid bilayer or to the proteins form the glycoproteins or liposaccharide. But these oligosaccharide or carbohydrate chain, as you see here, just located in the outer layer, not in the inner layer. And they give a asymmetric structure to lipid layer, and also they form a shell of the carbohydrate in the outer part of the cell membrane that named glycocalyx. Glycocalyx is the shell of the carbohydrate in the outer part of the cell membrane that involved in the cell recognition, cell adhesion, cell identification, and other function. The other component that is in the cell membrane and interact with the phospholipid bilayer are cholesterol molecules. Cholesterol stabilize the lipid bilayer but have two opposite functions. In some part, it restricts the movement of the lipid bilayer, but in the other hand, it can modulate the fluidity of the cell membrane and also can restrict the movement of some proteins in the cell membrane. Cholesterol give a fluidity to the cell membrane that this model named fluid mosaic model that some membrane proteins that not bound rigidity to the phospholipid and not bound rigidity in their place and can able to move in the cell membrane. In vitro condition, after fusion of one cell with another cell that labeled with fluorescent dye, after some passing of some time, these test cells fuse together and the dye spread in the whole of the cell membrane. That is show that some proteins can move in the cell membrane. That this is a function of the cholesterol that give the fluidity to the cell membrane. But in the other hand, cholesterol can restrict the movement of some proteins, some membrane proteins that involved in the cell signal. These type of proteins must not move in the cell membrane. In the lipid raft structures that include the high concentration of the cholesterol and also saturated fatty acid, these lipid rafts restrict the movement of some cell membrane proteins mostly involved in the cell signaling. That these structures located in some part of the cell membrane or more in some cells that involve more in the signaling. Briefly, in this part, the part 1 of the cell membrane, the cell membrane consists of the lipid bilayer, including the cholesterol, and also the cell membrane include the lipid bilayer, that the cholesterol located between of the lipid bilayer. That they maintain the fluidity of the cell membrane and also can restrict the movement of some other membrane protein. Also, the lipid bilayer is in combination with membrane proteins such as integral and peripheral proteins that go across inside and outside of the membrane that involved in the membrane transporter. And in the outer part, they loosely attach to the cell membrane and can act as an enzyme that involved in the maintenance of the cell shape. You can study this part of the cell membrane from John Quira Basic Histology, Chapter 2.